Hi, I'm Peter Katsikas. I'm a professor of immunology at Erasmus University Medical Center in the Netherlands. As part of the ImmunAID project, we're also going to perform transcriptomic analysis of what we call microRNAs. Now, microRNAs are very small RNAs. Typically, RNAs are long. They have lots of little building blocks, if you want, which we call bases. They are hundreds or thousands long. But microRNAs are very small. They're around 22 bases only. It's only 22 little blocks. So they're very small, but they play a very important role in regulating how RNA actually works and how, from the RNA you can make proteins. So they're very critical in the function of the RNA, if you want. So what we want to do is to study the microRNAs in the populations of the blood. So what we do is we get blood from patients, we separate the cells which are important, which we believe are important in autoinflammatory syndromes, and then we analyze in each of these separate populations of cells all the expression of the microRNAs that they have. So in order to understand or to analyze the microRNAs in autoinflammatory syndrome patients, what we do is we get the blood from the patients. We use a special machine which is called a sorter, a FACS sorter, and this machine has the ability to separate populations from the blood, basically separates them into very pure populations. So you can separate what's an immune cell is called a macrophage from the blood. There's hundreds of different types of cells in the blood, and we can separate that specific population. And then we can get the RNA out of that cell, and we can sequence the RNA. And we do a special sequencing that only sequences small RNAs, that's where the microRNAs are. This is basically the technique that we apply to 4 different populations in the blood, which we will separate out with these machines. It's a lot of work to separate the cells, and then once you get the cells though and you get the RNA, then the sequencing gets a little bit easier, but then it's a lot of data analysis. That way we create a map, if you want, or a profile of the microRNAs in different immune cells. We think this is very important because immune cells, we believe, drive autoinflammation, and therefore having these maps maybe will give us clues to what is happening in the cells and maybe even give us also diagnostics.